One. All right. Hi, I'm Jake,
I'm Brianna.
and we're gonna be talking about the similarities and differences in gene expression between Alzheimer's and Huntington's disease patients. So neurodegenerative diseases occur when nerves in the brain or peripheral system lose function over time and ultimately die. A few neurodegenerative diseases include Alzheimer's, Huntington's disease, and Parkinson's disease. There is treatment as far as therapy goes that can help control some of the symptoms, but currently there is no cure or a way to slow down the progression of these diseases. Um, figure one, oh,
Oh,
sorry. In figure one here, it's basically a figure to show the brain of a normal, healthy individual versus someone with Alzheimer's disease. So you can, kind of visually see like the breakdown of, brain cells over time there. So, Alzheimer's disease is the most common form of dementia. It leads to destruction of memory and other behavioral functions. It causes atrophy of the brain, which is basically the loss of brain cells. It destroys a lot of cells that help a individual communicate. Huntington's disease, on the other hand, is very rare. It is a inherited disease, and it has a large impact on individual's function, abilities, as far as movement in the early stages, and because the progression can't be slowed down over time, an individual will develop cognitive disorders. So both of these diseases are progressive, incurable, and lead to the loss of cognitive function over time, and are caused by the destruction of brain cells. Um, Both of these diseases are caused by the improper assembly of proteins, which leads to protein deg- degradation. Sorry. Um, so, with what we know about both of these diseases, this kind of led us to ask the question: what are the similarities and differences between the two observed gene expressions in Alzheimer's patients and Huntington's disease patients? This is our research question, which is basically kind of the question I just asked. So, once again, when comparing the gene expression in Alzheimer's patients and Huntington's patients, what are the similarities and differences between the observed gene expressions, and how do they compare to gene expression in those of healthy individuals?
so for methods and materials, we looked at a lot of primary research articles, and that was to view general information about the two diseases and to find the SRA for the analysis. Cyverse is an online platform that holds millions of genetic coding files, and we used Cyverse to run an RMTA analysis to compare the genomes of the healthy and the sick individuals. And the RMTA analysis was a way of quickly processing the RNA data. BLAST is another online platform, and it allows you to compare nucleotide and/or protein sequences to determine the significant difference between the two. And we used RStudio, and that was used to run the feature counts analysis from the RMTA analysis. And we used it to create the volcano plots, which showed us the DEGs. So these were the results for Alzheimer's disease. There were 4 differentially expressed genes, and they were all downregulated. So 3 of the downregulated genes from Alzheimer's were small nuclear RNA, and the function of small nuclear RNA is it's a non-coding RNA that assists in modifying ribosomal RNA. And the last one was a far upstream element binding protein, which is a DNA binding protein that regulates the c-Myc gene, which regulates cellular metabolism and proliferation.
This figure shows the results of HG. It is a volcano plot from our studio analysis. It also shows how the genes express differently. There are 3 upregulated genes and there are 1 downregulated genes. Each of the genes have different function. For example, chromosome 21 likely contains 200 to 300 genes that provide instructions to proteins. Pseudogenes function regulates coding genes through the RNAi pathway. MicroRNA binds to and regulates expression of many other genes, sharing the RNA mechanism. X inactive specific transcript function encodes a long RNA molecule without any open reading frame. RNA products of XIST separate over and code the X chromosomes bearing the gene that produce them. In the Alzheimer analysis, all of the genes are downregulated, while in the HG analysis, most of the genes are upregulated, meaning increased expression of one or more genes. Only XIST is downregulated, and our paper is different from the paper that we read since they test blood protein while we focus more on genes that leads to different results. The paper that we read, there is no upregulation or downregulation of protein, no significant change in expression between disease. There is downregulation in the disease compared to the controls. The immune pathways were upregulated, RNA, DNA, and ATP were downregulated. Any questions? This is our work cited. Thank you.